Hey everyone, this is Michelle Montiel and Morgan Louvier, and we are from Grand Canyon University's Concept of Biochemistry course. And today we're going to be discussing the composition and function of nucleic acids. So first we're going to identify the main types of macromolecules. Then we will describe the structure and identify the function of nucleic acids. We're going to compare and contrast the structure and function of DNA and RNA. And then finally, the learner will analyze the effects of mutations and the function of nucleic acids. So let's go ahead and get started. So first, we're going to start off by discussing macromolecules. Do you know what macromolecules are? If you know, give me a thumbs up. And if you don't know, give me a thumbs down. Nelson and Cox say that the chemistry of living organisms is organized around carbon. And with that being said, there are 4 carbon-based biological macromolecules that are involved in the molecular organization of life. So those 4 macromolecules are carbohydrates, proteins, lipids, and nucleic acids. And these macromolecules are actually polymers of monomeric subunits that are involved in the structure of supramolecular complexes that make up the components of cells. So the first macromolecule we're going to discuss are carbohydrates. So carbohydrates are made up of simple sugars, which are also known as glucose. And they are used by the cells for quick available energy. And proteins, they are made of amino acids and they're used in DNA replication. They're also used in cell signaling, cellular transport. They speed up reactions and they form cells and tissue structures. Lipids are composed of fatty acids and glycerol, and they are used by the cell to make membrane structures and store energy. And finally, the last macromolecule we're going to talk about are nucleic acids. And nucleic acids are made up of nucleotides. So they store and pass on genetic information, whether that's in a cell or a virus. And there's going to be 2 types of nucleic acids, DNA and RNA. So like I mentioned on the previous slide, there are 2 types of nucleic acids, DNA and RNA. So they're each going to be made up of nucleotides that consist of a phosphate group, a 5-carbon sugar, and a nitrogenous base. The nitrogenous base, they're either classified as a purine or pyrimidine. The sugar in DNA and RNA are different though. So DNA contains the sugar deoxyribose, while the sugar in RNA is just simply a ribose. The nitrogenous bases are adenine, guanine, cytosine, tyrosine, and uracil. So they can be called A, G, C, and T. So A, G, C, and T are found in DNA, while A, G, C, and U are found on RNA. So each nucleic acid contains a phosphate sugar backbone, and the phosphate sugar residue is attached to the hydroxyl group on the 5-carbon of one sugar and the hydroxyl group of the 5' carbon of the sugar on the next nucleotide, which forms a 5-3 phosphodiester linkage. So the 2 types of nucleic acids differ in the amount of strands they are made up of. DNA is double-stranded while RNA is single-stranded. So if we look at the structure of DNA and RNA in this image, we can see the differences in the strands. So here is RNA if you follow my mouse, and it only has 1 strand, while DNA here on the right side is made up of 2 strands. So Erwin Chargaff, an Austrian-American chemist, actually looked at the structure of DNA and RNA, and he discovered that the nitrogenous base pairs are linked together in a 1-to-1 ratio. And if you can recall from the previous slides, there are 5 nucleotides that make up DNA and RNA. We have cytosine, guanine, adenine, thymine, and uracil. So Chargaff came up with the base pair rule that in a molecule of DNA, adenine always will bind with thymine, while cytosine always binds with guanine. And then in RNA, adenine will always bind with uracil, while cytosine and guanine will continue to bind with one another. So this complementary base pairing founded by Chargaff was essential to discovering the structure of DNA. And then if we look at the second image here, we know that this is DNA because it's double-stranded. We can see the two strands. And you can see that guanine is always going to pair with cytosine, and then we have our adenine and our thymine that bind. So now let's practice Chargaff's rule. So here we have a strand of DNA, and we need to determine what its complementary strand will read. So here's our strand if you look at my mouse. So the first nucleotide on the strand of DNA is adenine. So we have to think about what nucleotide binds with adenine. So if you thought thymine, that is correct. So continue writing the complementary strand in your notebook on your own. Okay, so now let's check your work. We know that adenine binds with thymine. So we have written our complementary base pair. And we have another adenine, so another thymine. Thymine will bind with adenine, so on. So then we get to guanine. So remember, guanine's base pair is cytosine. So you'll just continue to write the complementary base pairs until you have created the correct complementary strand of DNA.
Top. All right, so we had already gone over the composition of nucleic acids as far as the nitrogenous bases, the phosphate sugar backbone. Now we're going to go into what the function of nucleic acids are. And this we call the central dogma of molecular biology. That was termed by, I believe, James Watson. And so what this is, DNA is the instructions for life. So without DNA, we cannot really have RNA or eventually proteins. And so the central dogma means that everything stems from those instructions. So main functions of DNA, of course, are the storage of information in genes, which are small pieces of DNA. DNA is expressed in the form of RNA and eventually a protein. And then, of course, DNA is important to distinguish between species. That's what gives the instructions for the physical traits, the phenotypes, not only just the genotypes of different species. RNA is a smaller molecule. It's a close cousin of DNA, and it carries a message from DNA out of the nucleus because it is DNA is a big molecule, so it needs to create something smaller. So it carries a message from DNA out into the rest of the cell to eventually produce a protein. And remember, proteins are essential for structure, growth, repair, especially those special proteins called enzymes that speed up chemical reactions in the body. We will talk about transcription translation here in these other slides. So for right now, I would like for you to get with a table partner and go ahead and on your journal, on a sheet of paper, I want you to create this Venn diagram. So what I want you to do with all the information that we've covered, I would like for you to compare and contrast the characteristics and maybe functions or composition of DNA to RNA. I'll go ahead and give you 10 minutes to do that. So now that you've had the time to go ahead and compare and contrast, let's go ahead and go over what information you may have written down. All right, so we know that what they have in common, these nucleic acids have a phosphate sugar backbone. They have nitrogenous bases. The only difference is what kind they have. So for example, DNA has the sugar deoxyribose, RNA has the pentose sugar ribose. DNA is double-stranded, RNA is single-stranded. DNA has the nitrogenous bases adenine, thymine, guanine, cytosine, and adenine, thymine, cytosine, and guanine. And RNA has the bases adenine, uracil, cytosine, and guanine. And as far as structure, of course, DNA is longer strands and RNA is shorter strands. So if you have any differences there that from what I have on here, you can go ahead and correct whatever you have. All right, so let's talk about protein synthesis. I mentioned that DNA has to be changed into messenger RNA, or messenger RNA has to carry the message from DNA. We have several types of RNAs in the cell. Here, I'm just going to talk about protein synthesis, which is a main process that requires 2 steps to create a protein. And in a cell, if you look at the structure here, we have transcription and translation. Transcription is actually the message gets created from DNA. So we call that mRNA. So here's the message. It has to get created. It's a smaller molecule, and it goes through the nuclear pores in this case, and it has to be received by a ribosome. Ribosomes are organelles in which protein synthesis occurs. Here, we have another type of RNA called transfer RNA. So transfer RNA will bring over amino acids, which is represented by these red dots here, and it'll read the message on messenger RNA, bring over an amino acid or transfer it over, and then it'll drop it off and take off. Then rRNA, which is the third type of RNA, which is ribosomal RNA, will go ahead and assemble these amino acids. So pretty much here, it's translating the message into a protein. We don't go into a lot of detail as far as what enzymes. We kind of wanted to show you just the depiction that these are what's important as far as function. RNA is a big essential, or has an essential role in protein synthesis in 2 steps, transcription and translation. Now on paper, of course, we can practice this, and this is going to be very brief. So here, protein synthesis, I want you to try this. I'm going to show you how to do this. So it says transcribe the following DNA sequence to mRNA. We had talked about Irwin-Chargaff's rule, right? With DNA during replication, adenine will bind with thymine, guanine will bind with cytosine. Well, the rule changes a little bit when it comes to transcribing DNA into messenger RNA, or getting the complementary strand of RNA from DNA. So now the rule changes. Of course, when there's a T on DNA, there's going to be an A, just like before. But now if there's an A on DNA, there's going to be a U on RNA. So we notice the picture here. When this opens up, you're going to see U's on RNA. It has that nitrogenous base uracil. So here we have T on DNA, A would be on RNA. If A is on DNA, then U. C and G would stay the same. So if there's a C on DNA, it would be a G on RNA. If there's a G, it would be a C. All right, so this is your example here. Just remember, or if you want to learn this quicker, if there would have been a T there for DNA, it's going to be a U now. Okay, so go ahead and try this second example on your own journal or your own paper. And now, instead of making DNA, you're going to transcribe it or make RNA. So let's go ahead and check your work. All right, so here's the response. As you notice, there was a T on DNA, so I said there A, an A on DNA, there's a U, a C, there's a G, and so forth. If we keep looking here, if there's an A, okay, there's going to be a U. So this is transcription. So when you see U's, you're looking at RNA. You're not looking at DNA. That's the first step, right? Transcription. So now let's move on to translation. And really what it is, when we transcribe it into RNA, when it arrives at the ribosome, it gets read every 3 nucleotides, or every 3 nitrogenous bases. Every 3 is called a codon. So that's the code. This, what you see on the right, we call that a codon chart. So on paper, once we've transcribed the DNA, it has to be cut up into 3s, into codons. Then you use that codon to read the chart to find out what amino acid it codes for. What amino acid is that transfer RNA going to bring to the ribosome to start creating the protein? So here we have your examples. So AUG, we have already transcribed it, right? So then we're going to take AUG, and I'm going to translate it now. AUG, I look for my code. My first nucleotide would be here. Okay, my second nucleotide would be up here. And then my third one would be here to the right. So where the 3 nucleotides meet, or nitrogenous bases meet for that codon, that'll tell you what amino acid it codes for. So in this case, it codes for methionine. So we just, as shorthand, we would just write the first 3 letters, met. So then I move on to my next codon, ACG. So ACG, it's going to code for threonine or threonine. Then my next one, GGC. So GGC, I'm going to go here, G, and then GC, glycine. And then eventually there'll be a codon that tells the ribosome to stop making the protein. So in this case, we have UGA. So I'm going to read it here, UGA, and stop. At that point, the ribosome stops making the protein, no more transfer RNAs come in with amino acids, and the ribosome or rRNA starts assembling it, making sure everything's correct, and releases it out into the cytoplasm and or to wherever it needs to go. So then we have example 2. So here's example 2. We've cut it up into 3s already. So I want you to go ahead and use the codon chart and try to figure out which amino acids it codes for. So remember, we start with the first one, AUG, and you would write down the amino acid that it would code for. So go ahead and keep writing the amino acids. I'll give you a little bit of time there. So remember, you have to transcribe before you translate. Okay, so let's check your work. All right, so here we go. So we went ahead and translated the mRNA. So methionine, the next one is GGG. So we have here GGG for glycine. Then we have CCU, so CCU, proline, and then of course the last one, UAG, is stop. So you have several different codons that can code for those different amino acids. And that would be, remember that an amino acid sequence is a protein, or we call it a polypeptide. So we briefly wanted to kind of introduce that to you. We don't discuss as far as enzymes are concerned in the process, but this is just a brief introduction. All right, so after we've discussed the composition and the function of nucleic acids and of course how important they are, we do have a discussion question for you. So this one, it pretty much reviews or gives you a recap of the central dogma of biology. We would like for you to go and watch the video. And once you've seen it, I want you to think about how DNA, or we want you to think about how DNA is the instructions for RNA and eventually to create a protein. And the question here that we would like for you to answer says, how would a change in DNA sequence, which we call mutations, affect the processes mentioned in the video? Give 2 specific examples, citing at least 1 scholarly article to support your response. So after you watch that video, it's going to talk about some of the processes we mentioned in this PowerPoint. Let us know how mutations would affect this. And we have a digital wall we would like you to go to. It's going to be on Padlet.com. As soon as you go in there, you'll have a plus sign to go ahead and add your response. And we want you to go ahead and add your response there as well. And don't forget to cite your scholarly article in your response. All right, so any questions on the presentation, anything that you took away, any clarification you may need on any topics? I know we went into transcription translation. Just remember, those are just 2 processes that are included in protein synthesis. So it's a major function. We call that gene expression because a gene is a small piece of DNA and it has to get expressed into a protein, and that's the process that it follows. All right, so if there are no questions, then thank you for listening to our presentation. And as you can see, we do have our references here. So if you need to go and find more information where we got information, you can go ahead and visit those websites.